Uh, they're going to record it apparently, so for posterity. And I've known you for quite a few years, but tell us where you grew up.
Oh, I am actually from former Soviet Union. I was born in Kazakhstan. It used to be part of the Soviet Union, but I got all my education in Moscow, in Russia.
Yeah. And was that a prestigious place to get to and tough to get into?
Yeah, so in Soviet Union and now in Russia, Moscow has a big concentration of top universities, and I think that's why we all try to get education there. So I did my master degree in animal science, in mostly reproduction, and then I did a PhD in the Center for Medical Genetics. So mostly was interest in embryonic stem cells that time in a mouse.
Yeah. And was funding good at that time and when you were growing up and beginning as a young scientist?
It was okay. I think in graduate school, situation has changed with Soviet Union breaking up, and the situation was quite difficult with funding. So, but I was kind of right on the time when I finished before everything went down. So, um,
Yeah. And
but
then
before that,
after the fall of the Soviet Union, you moved eventually to the United States, to Utah, is that right?
right? Yeah, this was my first kind of postdoctoral fellowship training.
And what did you do there?
I came to the lab that was doing mostly, well, interested in cloning, of course, farm animals. And it was in 95. It was before kind of Dolly, before somatic cell nuclear transfer came.
Yeah.
And initially, we thought that you can only clone from embryonic cells. So they actually brought me because I had expertise in embryonic stem cells in the mouse. So I tried to establish embryonic cells, early embryonic stem cells in cattle and use those as a donor cells for nuclear transfer.
Yeah. Yeah. And you stayed there for about 3 years and then you moved, I think.
Yeah, so I moved to Portland, Oregon, where I joined Don Wolf. He was running a lab at the Primate Center. We have a Oregon National Primate Research Center, which primarily houses rhesus macaques. And the Don was also interested in cloning. It was actually in '98 when the somatic cell nuclear transfer was become available. So we were interested in cloning monkeys, but we also were interested in using nuclear transfer applications for clinical, because Don Wolf, he was my boss, and he actually was director of the IVF clinic here at the medical school. And in parallel, was running a research lab with monkeys.
Got it. And where in that timeline did you learn the technology of doing nuclear transfer, which is quite technically difficult?
Yeah, mostly it was at Utah State University because
Yeah.
we had access to large number of eggs. So the Utah State University is located in Logan, Utah, and where they have a huge slaughterhouse
Yeah.
cattle, and that's why we had multiple labs doing IVF in cattle because it was access to all sites,
Yeah.
including nuclear transfer.
Yeah. And now did you ever leave the primate center? I don't think you did you? You've been there ever since.
Yeah, so I've been ever since with Oregon Health Science University, and the Primate Center is a part of the OHSU. But I kind of moved from Primate Center a few years ago to
Downtown Portland, yeah.
downtown. Yeah, so mostly we work with human gametes and implantation embryos, and because of the complexity of regulations and funding, so we couldn't do much of this research at the Primate Center because it's a federal government funded facility. So we had to build the lab away from federally funded
Yeah.
buildings.
And we'll see that lovely building in your
Right.
first slide. And it's got a beautiful view over the river and downtown Portland, I think.
Yeah, this was one of the tall buildings that the
Yeah.
OHSU built a while ago, and it's a beautiful view so far.
Maybe before we get on with the talk, though, you might, not many people are that familiar with the National Regional Primate Centers. I've had the opportunity to visit two of them, the one in Portland and the one in San Antonio. They're absolutely remarkable facilities, aren't they?
Yeah, yeah. So we have right now, I think, 7 primate centers, and yeah, there is one in almost many states, and one in California as well.
Yeah. And how many animals do they hold there now? Oh, you're not allowed to say.
Oh no, I think it's between 5 to 6,000 rhesus macaques.
Yeah.
So rhesus macaques are predominant species at our primate center. Other primate center, they house a little bit different species.
Yeah.
But the rhesus macaques are most common.
And would you encourage people when they have the opportunity to reach out and do collaborative research with those centers?
Oh yes, of course. Each primate center was generated as a resource for entire community.
Yeah.
We have a small number of scientists, of course, but they are all working on their own projects. But primate center, of course, can provide particularly tissues and even animals on a collaborative basis.
Well, we really appreciate you joining us today, Shukrat, and very much looking forward to your talk. So thank you for being here.
Thank you. So let me find my presentation. Here you go. Can you see it?
Yep.
Yeah, so I'll be telling you a little bit about the nuclear transfer, because that's kind of my area of expertise, and particularly the nuclear transfer in reproduction. Oopsie. So nuclear transfer is by definition of transfer nuclear genetic material from one cell to another. There are actually natural forms of the nuclear transfer, and it's actually achieved by cell-to-cell fusion. For example, there are muscle fibers, a result of fusion of muscle cells, and it's become nucleated structure. But sometimes the cells could fuse to non-nucleated cell, and in that case, just will be one nucleus. Besides natural, of course, techniques of nuclear transfer, we use many artificial or experimental forms of cell fusion to achieve nuclear transfer. And I'll be telling particularly some of them in reproduction, particularly with oocytes and zygotes. So when two cells fuse, or of course, they could be from different origin, and that's why nucleus and cytoplasm could be the same origin or different, and it actually matters because as it forms in somatic cell nuclear transfer, you have a somatic cell nucleus, which will be transplanted into oocyte cytoplasm, and most of the time we actually, experts even in that field, have a hard time of calling that structure. Is it still a fibroblast, say, or it's an egg? And which is a very interesting question. So it's because it's a result of now reprogramming. As I mentioned, particularly in reproduction, there are forms of natural nuclear transfer. We consider fertilization as a form of nuclear transfer. In this case, both egg and sperm actually contain haploid number of chromosomes, and the fusion actually restores the normal ploidy. On top of the natural fertilization, we developed many forms of assisted fertilization. As you know, it's in vitro fertilization. We have now intracytoplasmic sperm injection, where we really do, technically the transfer of the sperm head inside of egg. So we developed also more advanced methods of nuclear transfer. Most of them are with eggs. We call spindle transfer. We could transfer polar body nucleus inside of egg. It's called polar body nuclear transfer. Pranuclear transfer, it's been known for a while. And lastly, somatic cell nuclear transfer. So I'll be reviewing some of the these techniques without going into technical details, but at least the applications and how we could be using them in clinical reproduction. So they mentioned the fertilization is natural form of nuclear transfer, but the IVF was actually developed for the same reason, so that naturally egg and sperm don't meet in the body, and that's why we have to recover it and mix them together. And natural IVF is just mixing sperm and egg, and the fertilization occurs naturally by fusion. But in many cases, this doesn't happen, and sometimes it's due to sperm motility or low number, and that's why the assisted fertilization is known as cytoplasmic sperm injection, or ICSI in short. So as you can see in this picture, we actually hold the egg, and then we pick up the sperm inside of tiny pipette, and we just directly inject into cytoplasm. So we break usually membrane. So we developed also a different form of nuclear transfer, which is called spindle transfer. This is actually transfer of the nuclear genetic material between two eggs. So I'll tell you why, but actually technically it's done to isolate nuclear genetic material in the egg. Eggs actually that we work with, it's mature oocytes. They in the metaphase of the second meiotic division, and that's why they don't have per se nucleus as an organelle. So DNA is already organized into meiotic spindle, as you could see, it's here. So it's just the chromosomes, and that's why instead of nuclear transfer, we call it spindle transfer. So there's some technical terms that we use. So when we do nuclear transfer, of course, first thing is to see that the nucleus, so you can isolate it. Unfortunately, with eggs at the metaphase 2, spindle is not visible. The pronuclei, would be visible nuclei because of the membrane, but here, because of the pure chromosomes, it's not visible. So we use very special imaging system, which is non-invasive, that allows you to see spindle proteins by fringes. And then we use tools to basically to go through zona pellucida and isolate this spindle inside of kind of membrane enclosed structure, which we call karyoplast. In this case, as you could see, the spindle is not really removed from inside of cell. It's actually encapsulated into membrane, and once it's isolated, you could see it's actually now round. So basically, this is enucleated eggs, and it's just cytoplasm, and the spindles are here. So the spindle by itself, and as a nucleus, cannot never be exposed to environment. So it has to always sit within cytoplasm. That's its natural environment, and so that's the way we keep it, even though we call it nuclear transfer. But in reality, this nuclear genetic material is actually small cell. So once you have this separation of the nuclear genetic material and cytoplasmic, now, of course, we can take this spindle and put it inside of different egg. And then this fusion is done by more or less natural, membrane fusion techniques. So we very much use natural ways of cells, do nuclear transfer, and we try to basically mimic most of the techniques. So that's actually how nuclear transfer works. So it's not really transferring pure nucleus, but it's more like using cell division. We basically divide egg into two using very similar techniques, using the cell membrane to isolate them into two. So one of the reasons why you would do transfer of the spindle from one egg to another is because of the mitochondrial content. As you could see, the mitochondrial DNA or mitochondria is a cytoplasmic organelles, and they located in the cytoplasm, where the spindle, if you isolate, would have mostly, of course, chromosomes with maybe very little amount of cytoplasm containing mitochondria. So basically, here you could see we actually uncoupled nuclear mitochondrial DNA, and that way we could actually retain the nucleus, but replace the cytoplasm from, for example, from different patient and with aim of replacing mitochondrial DNA. The mitochondrial DNA, even it's a tiny genome, it's the mutations in this genome have been implicated in many devastating conditions in humans. So it's because of the mitochondria primarily or predominantly inherited through maternal lineages, mostly through egg. So that's why women would pass her mtDNA mutations to all children. There are actually cases when not every egg will contain the same load of mutation, because most of the mutations I have to mention in mitochondrial DNA, they kind of heteroplasmic, because there is so many thousand copies of mtDNA in each cell, particularly in oocyte. Not all of them become mutant. It's usually a mixture of mutant and normal wild type, which is called heteroplasmy. And that mutation load can be different from one egg to another, but most of the time, almost every egg will contain mutation. But the question is whether it's cause severe disease or mild. So for these families, usually they have a mitochondrial disease. Of course, the father wouldn't pass unless there are very rare cases when paternally inherited mitochondria can be found in children, and sometimes in very high heteroplasmic cases. So for families that carry mutations, maternally inherited mutations, unfortunately, the PGD doesn't work really well, just because if we would create multiple embryos from several eggs, and then we would do biopsy, so most of them would contain mtDNA mutations, but we would have to select with least mutation and transfer, which still could cause the disease. So for these families, we developed this mitochondrial replacement where we take, as I mentioned, the spindle from a patient, remove it, and then transplant into cytoplasm of another egg derived from a donor. So we actually developed this procedure initially in rhesus macaques. So most of the difficulties with transferring spindle were resolved early. And in rhesus macaques, we actually, because we didn't have any model with mtDNA mutations, mostly we used healthy macaques, but different origin. As we discussed, we have rhesus macaques, primarily the primate center, but we have two kind of subspecies. It's Indian and Chinese origin, and they quite genetically different, particularly, for experts who's working with rhesus macaques, we always know, how different they respond to treatments. But anyway, so we use these two different macaques to replace the mitochondria. So we would take Chinese origin macaques mitochondria, but the spindle would come from Indian or vice versa. So using this technique, we produced several macaques in 2009. And despite of this, the mitochondrial replacement, what we call it, these macaques were pretty normal, even though, we can measure differences in mitochondrial sequence between, donor and recipient. And so because mitochondrial genome is quite small, so mostly we can detect the SNP differences. So we actually found that Indian and Chinese macaque differences are much higher than, for example, within human population. For example, we found that rhesus macaque of Chinese and Indian origin may differ in the mtDNA, by just SNP numbers is up to 700 to 800 SNPs, where the humans maximum known is 100 SNPs. So as you could see, the humans have much less difference in mtDNA. But despite that, this was quite unremarkable for rhesus macaque, and we looked at the development and aging. Right now, they actually all the adults. But because we developed this procedure for humans, so we looked at many safety and efficacy considerations. First of all, as I mentioned, when we do this transfer, it's not absolute, so because the spindle comes with tiny cytoplasm, as I mentioned. This is the must-have. We cannot just clean up and rinse spindles. So unfortunately, it comes with tiny cytoplasm, and that means with a mitochondria that bound to this to membrane or a spindle. Which is called carryover, but usually it's a very small amount. By our estimates, it's about 1% of mitochondria now in newly created egg will be maternal. It's called carryover. And that means the other 99% are donor. So that's unfortunately impossible to remove it completely. And so this carryover amount is always there. And of course, in this case, if there was a mutation in the egg, so we say, we remove 99% mutation or replace, but 1% remains. The other thing is, how, this 1% will remain, because, initially it's a 1%, but during development, the replication, this heteroplasmy could change. So and I'll show you later, we actually found that because of the mysterious mtDNA replication, sometimes even this tiny amount of mtDNA from a mother could actually increase significantly in a baby. The other the issue that the regulatory agency has been asking us when we brought this, for approval is how much donor mtDNA should be similar or different from original material. As I mentioned, every humans will contain their own haplotype, haplogroup, slightly different from each other, but there are still differences, particularly because of geographic origin. And up until now, it's not clear whether the donor mitochondrial genome has to be matched as close as possible to original material or not, or any random selected egg donor could be used. This question is because of the mitochondrial and nuclear genome are very closely associated. As you know, the mitochondrial genome encodes OXPHOS proteins, but only part of them, because actually majority of them, only 13 proteins come from mitochondrial genome, where the rest, about 100 total, they come actually from nucleus. So as you can see, nucleus and mitochondria co-encode complexes, and there is a suggestion that because of the slightly differences in protein, that they may not actually form normal complexes, which will result in some kind of mismatch. This actually happens mostly with interspecies differences. But within species, it still hasn't been really shown to be a problem. And so far, it doesn't seem like within species, looks like any mtDNA would work for this procedure. And also, I think there was the suggestions that we actually match exactly, and so in this case, the exact match is hard to find. It has to be probably the relatives of the family, but normally its relatives should be maternal lineage, and since this mutation comes with maternal lineage, we often cannot use any maternal relatives as the donors. So anyway, in rhesus macaques, we developed this procedure just to see how much of these mtDNA sequence differences could affect, and many of these macaques were created with Chinese Indian combinations were quite different, as I mentioned, than humans, so almost tenfold difference in terms of genetic distance. And that doesn't seem like mattered. Of course, it's in the same species, and we know Chinese and rhesus macaques can breed normally without any problem. But here we actually showed that this mitoid tracker, our firstborn in 98 male macaques, they now middle-aged adults, and they fertile. There are some suggestions that maybe this mismatch will affect the fertility. So this is actually the offspring. This is Mitas daughter, yes, and this is tracker. In terms of females, we had one female MRT macaque, and so we bred her with, naturally with male, and this is actually her offspring. And in this case, of course, because her mom carried donor mitochondria, and now she passed it to her offspring. So it's a kind of germline transmission of the donor mitochondria now to next generation. And other than that, I think we looked at the health-wise, the developmental milestones, and they were so far unremarkable. These macaques were as normal as any other that we've seen at the primate center. So I mentioned about this phenomenon that when we create this spindle transfer oocyte, only about 99% would be from donor, which is healthy, where the small amount of mtDNA copies will come with a spindle because it's, as I mentioned, surrounded with cytoplasm. And this is, called heteroplasmy. So there's a 1% possibility of carrying over mutation from mother. We always thought that the 1% mutation is not going to affect the disease, which is true. So usually the disease will strike when mutation reaches more than 60% in giving cell. So 1% shouldn't be, detrimental. But unfortunately, what we found is that if you, particularly in humans, if you develop these spindle transfer oocyte, you fertilize them into embryonic stem cells. So what we found is some embryonic stem cells gradually show increase in this maternal mtDNA. So if you measure it, it becomes, from 1% to 5, 10, and some cell lines within passage 10 would completely reverse back to maternal, or like, we never actually did a replacement. So this phenomenon is called the reversal, and it's been found in mostly with human embryonic stem cells, not with children yet. But in any way, so this is a very interesting phenomenon that we still don't understand. How could these tiny number of mtDNA copies that we introduced with a spindle could have a replicative advantage. So during development, this could actually increase. Fortunately, this of course doesn't happen very often. So far, it's unclear, how frequently it happens, but our estimate is about 20%. So in 20% cases, you may have this type of reversal. So with embryonic stem cells, these reversals we've seen actually complete. It's very quickly would switch to completely to 100% maternal with loss of donor. But in reality, at least with some animals, we see that this is kind of partial reversal, where there's increase from 1% could be up to 50%. But so far, it's never been completely losing of healthy donor mtDNA. So because of the regulations here in the US, unfortunately, we haven't been able to test these techniques directly with patients. We did it with eggs and embryos, embryonic stem cells, without transplanting them. But because of the IVF is a kind of mostly privately run clinics enterprise, so many IVF clinics outside of United States now using this technique. This is actually was US clinic that took these patients into, I think the clinic, secondary clinic in Mexico, and they did treatment for this family. This family was diagnosed with maternally inherited mitochondrial mutations causing Leigh syndrome, one of the devastating syndromes. And they did MRT for these patients, and so far the child was born healthy. So there is a slight increase, I guess, the mutation load, in skin, actually, in fibroblasts, as you could see, up to 9%, which is, still well below threshold of, expression of the mutation. But in any way, despite the controversy, because it hasn't been done in a regulated way, but this is considered to be one of the first cases of germline gene therapy by mitochondrial replacement. So some of the highlights of this spindle transfer we call mitochondrial replacement therapy. First of all, it's a use of mitochondrial genome from donor. So it's not recombinant, it's not really gene correction. So it depends on donating by somebody egg, so we could use mitochondria. Unfortunately, only eggs of the same stage has to be used for some technical reasons, so it has to be egg. Another thing is because of so many different mutations in this mtDNA molecule, we have different diseases, but since this treatment kind of replaces whole genome, so it's applicable to any mtDNA mutation, whether it's a Leigh disease or whether it's other syndromes caused by mitochondrial mutations or deletions. As I mentioned, it's preclinical studies been done in macaques for almost now. We have second generation macaques, and so far they show efficacy and safety. Except of course some of the cases is reversal, which is still the ongoing, and we still doesn't seem like understand the biology or basic biology of mtDNA replication to prevent or somehow to affect these cases of reversal. So this spindle transfer mitochondrial replacement is also whole cytoplasm replacement, as you could see. So we've been interested in using it not to kind of treat mitochondrial DNA mutations, but also just to replace mitochondria, and when replace